Hello, my name is Sarah Cox-Vazquez, and I'm a recent Rams Lab alumni from the University of Michigan. Today I'm going to be telling you a little bit about the biophysical characterization of amyloids that happens in the Rams Lab. Protein misfolding diseases, or amyloid diseases, are still quite poorly understood. Some of the most common ones include Alzheimer's disease, type 2 diabetes, cataracts, prion diseases, and rheumatoid arthritis. And what all these multiple diseases have in common is that they all have something called amyloid fibers, which are really long fibers consisting of multiple repeat units of the same proteins over and over again to form these really long fibers that are very, very stable. While all of those diseases are in different parts of the body and their normal protein functions are very different, there are some unifying characteristics that make them amyloid proteins. For example, amyloid fibers consist in a very low energy minima, which makes them very stable to any type of degradation, including temperature changes, pH, and proteases. They also form a very characteristic beta sheet amyloid fiber, in which repeating units of the same protein are stacked on top of each other, almost like a deck of cards, and then form very long fibers perpendicular to the axis. They form extracellular deposits outside of the cells, which they can cause damage and act as nucleating sites for further amyloid growth. And finally, they follow a very similar nucleated polymerization mechanism in which monomers of the protein then become fibers. One of the most famous amyloids is amyloid beta, which is known as a contributing factor to Alzheimer's disease. It is cleaved in the extracellular membrane of neurons by beta and gamma secretase from amyloid precursor protein. In a healthy state, it's also cleaved by alpha secretase in its amyloidogenic core, at the residues highlighted in red, which are known to be the nucleated sequences for the amyloid beta fibers in this disease. The Alzheimer's brain is characterized by a general shrinking of the cortex and hippocampus, which are areas that are devoted to memory. And on the cellular level, it's characterized by both internal and extracellular amyloid plaques. All amyloids follow a very similar aggregation mechanism, in which the protein or peptide starts as a single monomer and eventually interacts with itself to form multiple types of oligomers, before eventually reaching the protofiber stage, in which the nucleated sequence has come together to form the fiber core, before finally forming these very hyperstable and very long megastructures of fibers. And while amyloid fibers are the most commonly known portion of amyloid diseases, it's actually these oligomers, which are heterogeneous and transient, which are thought to be the toxic species in amyloid diseases. The Ramamoorthy lab, or Rams lab, uses a variety of biophysical tools to investigate amyloids. For example, we use a lot of NMR and circular dichroism to investigate the structure of the amyloids. We use a lot of fluorescent assays such as Thioflavin-T, membrane permeability, cell viability, to understand how the amyloids are interacting with cells and what their aggregation mechanisms are and how they can perturb membranes. And we also use a lot of TEM, transmission electron microscopy, to look at the morphology, as well as lots of chromatography, molecular dynamics, and many more techniques. Both one-dimensional and two-dimensional NMR are very useful for studying amyloid proteins. For example, one-dimensional proton NMR is useful for understanding the overall solubility or level of aggregation of the amyloid in solution. And 2D NMR is useful to be able to probe each individual residue and how it's affected by ligands or membranes, for example. An NMR is very useful because the intensity of the NMR peak can be correlated to the size of the amyloid in solution. For example, a monomer in solution will spin very fast and it will give a very sharp peak, whereas when it begins to aggregate, it'll spin more slowly in solution and the peak will start to become more broad. And once it's reached its full fiber state, the tumbling is so slow in solution that you no longer can detect it via NMR. Here we show an example of SOFAST HMQC two-dimensional NMR, in which we were investigating how 5 different ligands affect the overall amyloid beta aggregation. And we were able to determine how the ligands were able to inhibit the aggregation or speed up the aggregation of amyloid beta using two-dimensional NMR. Another common assay is the Thioflavin-T assay, or THT assay, in which we use a fluorescent dye which only becomes fluorescent when it binds to the beta sheet structure of the amyloid fibers, which is very useful for helping us to track the kinetics of amyloid formation. Here I show an example of 21 different compounds and their impact on amyloid aggregation using the Thioflavin-T assay, in which we are able to investigate what compounds fully inhibit aggregation, what compounds speed up aggregation, or even how they disaggregate fully mature amyloid fibers. We also use transmission electron microscopy, or TEM, to look at the different morphology of the aggregates and the amyloid fibers. Here we show how a variety of different compounds can greatly alter the fiber morphology through either inhibition or promotion or generating just different types of fibers in general. We can also use circular dichroism spectroscopy to understand the overall fold of the amyloid peptide or fiber in solution. For example, amyloid beta as a monomer exists as a random coil structure, meaning it's generally unstructured, while the amyloid beta fibers are highly structured and consist in a beta sheet formation, meaning that they have this negative peak around 215 nanometers. Here we show how we can use circular dichroism to monitor the secondary structure changes of amyloid beta in the presence of 5 different compounds. So these were some of the ways that we use biophysics to investigate amyloids in the Ramamoorthy lab. And this was just a very brief overview. I invite you to scan this QR code here to check out our amyloid publications. Thank you so much for your time.